Okay, so moving on to section 3.2, which is the internal structure of the cell. I'm going to cover this very briefly and in minimal detail. This is important information, but there isn't time to cover all this information in depth. I think it's just a little too much for a 200 level class. So I'm gonna, in my slides, I'll give you the information that I think is most important. Please don't stress about knowing much more than what's on the slide. Okay, so first, the ions that are most critical to neuronal communication, the ones that might come up in later chapters, are sodium, potassium, calcium, and chloride. You don't need to know much more than that right now. The cell membrane is rather important, and we'll talk about it a bit in the next chapter as well. It's the double-layered outer wall of the cell, and it regulates what enters and leaves the cell. So proteins, there are proteins that are embedded in the cell membrane, and they act as kind of like gatekeepers to decide what goes in and out of the cell. And the cell membrane, the double layer, is made up of phospholipids. So one layer is hydrophilic, and one layer is hydrophobic. Hydrophilic literally means loves water, and hydrophobic means hates water. The hydrophobic part is made of fat, which you know resists water. The nucleus of the cell, which is located in the soma, is kind of like the executive office of the cell. It contains DNA, which is the blueprints for making proteins, chromosomes, which are double-stranded molecules of DNA, and then genes, segments of this DNA. And there's a nice picture up here, and then one down here as well to show that the relationship between chromosomes, DNA, and genes. And then importantly, nucleotides are the building blocks that make up DNA, adenine, thymine, guanine, and cytosine, which are typically referred to or abbreviated as just ATGC. And just some brief information about proteins for anyone that might have forgotten from the last time they took biology. They are folded up polypeptide chains that serve a particular function in the body based on their shape, their structure, and they can change shape and combine with other proteins. They can be transported from one neuron to another through the axon. Proteins embedded in the cell membrane, as I mentioned just before when we were talking about the cell membrane, form passageways or gates, and these are called, they're called channels, gates, and pumps, and they regulate the flow of substances in and out. There's also a receptor site, as you can see up here, which is like a little groove in the protein, and they're sort of analogous to a keyhole. So if the right molecule comes in, then it might change the shape of the protein. Okay, so moving on to section 3, genetics. Important that you know the difference between a genotype and a phenotype. The genotype is the genetic makeup of an individual, so the actual genes that you're born with. And a phenotype is the observable physical and behavioral characteristics of an individual. And these results from the interplay between the genes and the environment. We're going to talk in this chapter mostly about Mendelian genetics. It's the basic laws of genetics, but it's not the whole story. And you might remember that Gregor Mendel is the father of the science of genetics. He was kind of the first person that described genetics in the way that we basically understand it now. He was this, I think, Czech monk who was really into his pea plants. Okay, so some basics of genetics. The nucleus in each human somatic cell contains 23 pairs of chromosomes, so 46 chromosomes in all. The chromosome pairs are numbered 1 through 23. Autosomes are is what we call the chromosomes in pairs 1 through 22, and then the 23rd pair are sex chromosomes, so that's the X and the Y. And typically males have XX chromosomes and females have XY, but there are, of course, deviations to this. One member of each pair, so one chromosome from each pair comes from the mother, and one chromosome of each pair comes from the father. An allele is a version of a gene, and a gene pair contains two different alleles. If both alleles in a chromosome pair are the same version of that gene, they're homozygous. And if those alleles in the pair are different, they're called heterozygous. Alleles are expressed in different patterns. One pattern is complete dominance, where only dominant gene is expressed and the recessive gene is completely suppressed. Incomplete dominance is when each allele's trait is only expressed partially, and codominance is where each allele's trait is expressed fully. Okay, genetic mutations. These are errors in copies of a gene that create altered alleles. So it can be a change in a single nucleotide, for example. So one of these A, T, G, or C nucleotides, and that creates a new single nucleotide polymorphism. Genetic mutations can be a blessing or they can be bad. So there's the hemoglobin mutation that causes sickle cell anemia is bad, but then a different mutation or a different version of this gene can be good. It actually offers resistance to malaria. Okay, so one example of a genetic disorder that affects the brain, we'll talk about a recessive one and a dominant genetic disorder. So the recessive, an example of a recessive genetic disorder is Tay-Sachs disease. And this is, in this case, a dysfunctional protein causes buildup of fats in the brain, and the individual dies, I think, within 5 years of life at maximum, typically. A baby who is homozygous for recessive mutation of this gene will have severe motor and cognitive impairments. Because this gene is recessive, both parents have to be carriers. And if both parents are carriers, if you look at this figure on the right, there's a 25% chance that the offspring will inherit this disease. Okay, so if you see over here. Carriers for the disease, interestingly, have protection against tuberculosis. So if you have one dominant and one recessive allele, which means you would not have the disease, but you would be a carrier, there's kind of an evolutionary advantage to being a carrier, which might explain why this disease is still present in our gene pool. An example of a genetic disorder that is dominant is Huntington's. So most of you are probably familiar with this. It's caused by an abnormal version of the Huntington protein, which kills brain cells, especially in the basal ganglia and the cortex. If you remember, the basal ganglia has a range of functions. The Huntington's disease, one thing you can observe that's kind of marked is this chorea, which means like dancing, and it's an involuntary movement of the limbs, and you can look at this YouTube video of an individual who has Huntington's chorea. The allele for this gene, for this disease is dominant. And if you look over on the right here, you'll see that that means that offspring who have it, offspring with one parent who has it, there's a 50% chance of getting the disease. If there are two parents who carry the dominant gene, there is a 75% chance of inheriting the disease. But notably, only one copy is required to inherit Huntington's disease. Okay, moving on to chromosomal abnormalities. There are two main ways that chromosomes can be abnormal. One is a deviation in the copy number variance. So too many or not enough copies of a disease can cause too many or not enough copies of a gene can cause issues. Several psychological disorders, such as autism, schizophrenia, and some learning disabilities, are associated with copy number variance, changes in copy number of part of a chromosome. An entire chromosome can also either be transmitted as a pair, as a double, or not enough. So Down syndrome is an example of a situation where there's one extra chromosome. So the 21st chromosome is copied, I think typically on the mother's side, and it causes a range of issues. Probably everyone is familiar with some of the cognitive problems, motor problems, and cardiovascular issues associated with Down syndrome. Quick note, don't worry about the section on genetic engineering. It is definitely interesting, but it's not directly applicable to things we'll talk about later, so you can read it if you're interested, but don't worry about studying that part. Okay, so all this talk of genes, it's important to note that genes alone do not determine a person's phenotype. Otherwise, two people with the same genes, such as identical twins, would look and act exactly the same. So some examples from my personal life: I have an identical twin sister. She's here on the right, and she and I have very different phenotypes, both behaviorally and physically. We've always looked very different, and we also have quite different preferences. She's an artisan cheesemaker, and she lives on a farm and is great at cooking, and I am obviously in academia, and I am terrible at cooking, so we're quite different. And then I couldn't resist, here's another example. These are my two baby nieces. They are identical twins as well, and even at 7 months old, they're already quite different. So Lucy over here likes carrots, and she likes reading books, while Eva does not like carrots, and she likes playing with her big brother. And they also look rather different. They look different enough that that you can tell the difference between the two. So even after 7 months, the environment has interacted with their genetics enough to differentiate the two. So phenotypic plasticity is the ability of the genotype to produce more than one phenotype. Epigenetics is the fact that an organism's environment can change how a gene is expressed. This doesn't mean that the actual genes are changed. There is kind of a next level of chemicals that can turn a gene on and off. And this is another way in which the environment affects our phenotypes. There's been a few cases in which scientists have noted that we can kind of inherit some epigenetics. So organisms might inherit some of these we call them epigenetic tags from their parents, which affect how the genes are expressed. Again, the genes aren't actually changed, but they're kind of tagged by certain chemicals that turn them on and off, and so some of this can be passed through from parent to offspring. And this has been likened to inheriting the experience of your parents. One notable example is that in Israel, they did a study where they found that Holocaust survivors may have passed on susceptibility to stress due to epigenetic changes during their traumatic childhood. So these were Holocaust survivors who had been in concentration camps and, of course, had extremely traumatic childhood experiences. This changed their own epigenetics. It changed the way that the Holocaust survivors' genes were expressed. And there's evidence that they passed some of these epigenetic tags on to their offspring, such that some of the children of Holocaust survivors have a higher risk for developing PTSD or a higher susceptibility to stress. Notably, there have been some studies that also indicate that while some children of Holocaust survivors have a decreased resilience to stress, some also have increased resilience. So it's a relatively new field of study, and hopefully some more clarity will come over time.